The ovaries are among the first major organs in the human body to show a clear age-related loss of function. Long before the heart, lungs, or brain reach anything resembling complete failure, the ovarian reserve is already declining. That process eventually leads to menopause, but scientists still have very few proven ways to influence how quickly ovarian aging happens. A new Nature aging study now points to something surprisingly familiar: physical activity. In more than 150,000 participants from the UK Biobank, lower physical activity was associated with being postmenopausal, and a second analysis of more than 12,000 participants from Haines showed a similar pattern. But the more interesting part came from mice, where researchers could actually test cause and mechanism. Exercise delayed several features of ovarian aging, and much of that protection appeared to depend on a hormone called adiponectin. Exercise is already associated with better cardiovascular health, stronger muscles, improved glucose control, and lower risk of many chronic diseases. Why would the ovaries be different? The important question is not whether exercise is generally healthy. It is whether physical activity can directly change the biological machinery that controls ovarian aging. That is what the researchers tried to investigate. Their study combined large human datasets with controlled animal experiments, allowing them to separate an interesting population-level association from a mechanism that could actually be manipulated in the laboratory. The researchers analyzed 152,435 participants in the UK Biobank and compared physical activity levels between premenopausal and postmenopausal individuals. They found that lower physical activity was more common in the postmenopausal group. They then examined another 12,418 participants in the U.S. National Health and Nutrition Examination Survey and found supporting evidence for the same association. These are large data sets, but there is an important limitation. Both analyses were cross-sectional. They show that activity level and menopausal status are related, but they cannot prove that exercising more directly delays menopause in humans. It is also possible that menopause, health status, body composition, or other lifestyle factors influence physical activity. That is why the mouse experiments matter. In animals, researchers can control age, environment, exercise, genetics, and treatment far more precisely. When female mice were given higher levels of physical activity, the researchers found evidence that ovarian aging slowed. The exercised animals preserved more of their ovarian follicle reserve, the population of follicles containing immature eggs that progressively declines throughout reproductive life. Once that reserve becomes severely depleted, fertility falls and the reproductive lifespan approaches its end. Adiponectin is usually discussed as a metabolic hormone produced by fat tissue. It is associated with insulin sensitivity, energy regulation, inflammation, and communication between metabolic organs. Yet the new work suggests that adiponectin may also act locally inside the ovary and participate in the way physical activity protects reproductive tissue. The researchers then performed one of the most important experiments in the study. They reduced adiponectin specifically and asked whether exercise would still protect the ovarian reserve. The answer was only partly. When adiponectin was deficient, the beneficial effect of physical activity was attenuated. Exercise was no longer able to produce the same level of protection. That does not mean adiponectin explains every benefit of exercise, but it strongly suggests that it is one of the signaling pathways connecting physical activity to ovarian aging. Muscles are not just mechanical structures that burn calories when they contract. Physical activity creates signals that travel throughout the body. Hormones, metabolites, inflammatory molecules, and growth factors change in response to movement. The ovary appears to be listening to some of those signals. In this study, adiponectin became one of the messengers carrying information from a physically active state into reproductive tissue. They used AdipoRon, a compound that activates adiponectin receptors, in mice, AdipoRon delayed features of ovarian aging, preserved follicle reserve, and extended reproductive lifespan. This is important mechanistically because it moves the study beyond simple correlation. Exercise increased adiponectin, loss of adiponectin weakened the benefit, and stimulating adiponectin receptors reproduced part of the protective effect. Together, those experiments form a much stronger causal chain than the human observational data alone. It plays major roles in cell growth and metabolism, but excessive or poorly regulated mTOR activity has also been implicated in many aspects of aging. The extended data from the study show that adiponectin manipulation altered ovarian mTOR signaling, and previous work has shown that suppressing mTOR can influence activation of primordial follicles. That matters because follicles represent a limited resource. If too many dormant follicles are activated too quickly, the ovarian reserve can be depleted faster. Imagine the ovarian reserve as a savings account that cannot be refilled easily. Each month, follicles are recruited from that reserve, but only a tiny fraction will ever lead to ovulation. The rest are lost through normal biological processes. If the rate of recruitment or follicle loss becomes too high, the account runs down faster. One possible interpretation of the new research is that exercise-associated adiponectin signaling helps create a metabolic environment that protects more of that reserve and slows some of the processes that push ovarian tissue toward aging. The researchers also examined markers related to apoptosis, the controlled death of cells. Their extended data included measurements of proteins such as BAX, BCL2, cleaved caspase 3, and tunal staining, all commonly used to study cell death. Exercise and adiponectin-related interventions affected these pathways, supporting the idea that the mechanism may involve protecting ovarian cells from age-related loss rather than simply changing circulating hormone levels. This is where the story becomes especially relevant beyond fertility. The ovary is an endocrine organ. As ovarian function declines, levels and patterns of reproductive hormones change dramatically. Menopause is therefore not simply the moment when pregnancy becomes impossible. It represents a major systemic transition that affects bone, cardiovascular physiology, metabolism, and other tissues. The authors frame ovarian aging as a process connected not only to infertility, but also to endocrine dysregulation and broader chronic disease risk. But there is an obvious danger in turning these results into a headline such as "Exercise Delays Menopause." The human part of the study does not establish that. A woman cannot use these findings to calculate how many extra reproductive years she will gain from running, cycling, or walking. The study also does not identify an optimal exercise dose for preserving human fertility. Very high levels of exercise, especially when combined with inadequate energy intake, can disrupt reproductive hormones and menstrual function. So the message is not that more exercise is always better for the ovaries. The AdipoRon result also needs caution. AdipoRon extended reproductive lifespan in mice, not women. It is not an approved anti-aging treatment for the human ovary, and this study does not demonstrate that taking an adiponectin receptor agonist would safely delay menopause in people. Signaling pathways such as adiponectin and mTOR operate throughout the body, so manipulating them pharmacologically could have effects far beyond reproductive tissue. The authors themselves present adiponectin as a potential therapeutic target, not as an established therapy. There is another reason the study is interesting. It provides a potential explanation for why physical activity could affect reproductive aging even when body weight does not dramatically change. In the mouse experiments, the researchers examined body and ovarian weight alongside reproductive and hormonal measures. The protective mechanism was not presented simply as an effect of making the animals thinner. Instead, the experiments point towards signaling changes inside the ovary itself. Exercise is a whole-body signaling event. Contracting muscle changes metabolites and hormones. Fat tissue changes its secretory behavior. Immune cells respond. Mitochondria adapt. The brain changes. And according to this new study, the ovary may be another organ receiving those systemic messages. The most valuable next step would be longitudinal human research. Instead of comparing active and inactive people at one point in time, researchers would need to follow women for years, objectively measure physical activity, track ovarian reserve markers, reproductive hormone changes, menopause timing, and potential confounding factors. Randomized exercise trials could test whether increasing activity changes biomarkers such as anti-Mullerian hormone or antral follicle count, although proving an effect on the actual age of menopause would require much longer studies. Researchers will also need to determine whether adiponectin plays the same role in human ovarian tissue that it does in mice. The animal experiments make the pathway biologically plausible, but humans and mice differ substantially in reproductive lifespan and follicle dynamics. A treatment that extends reproductive life in a mouse may produce a much smaller effect, no effect, or a different effect entirely in humans. Still, the study offers an unusually coherent chain of evidence. Large human datasets show an association between higher physical activity and reproductive status. Controlled mouse experiments show that exercise can preserve ovarian function. Exercise increases ovarian adiponectin. Removing adiponectin weakens the effect. Activating adiponectin receptors recreates part of it, and downstream metabolic pathways such as mTOR provide a plausible connection to follicle preservation. And it does not mean scientists have found a drug that can safely extend human reproductive life, but it changes one important assumption. And one of those signals appears to become stronger when the body moves.